Apis Rescue, we're a nonprofit. We're actually a classified 501c3 nonprofit. We're looking at the genetic diversity in feral populations is one of the main things that we're looking at. These are the tenets that we follow when we're looking at that. Research, of course, but also we're really interested in education, getting good education out to people. That's the whole reason for this conference, is a better education and community, because community is important. And then we would like to bridge the gap between beekeepers and research. A lot of times research is technical jargon that is not accessible to your local beekeeper. It's not easily digestible. We want to bridge that gap. So have people who are trained on both sides so that we can make the information that's coming out, the newest and best information when it comes to fighting varroa or diseases, make that more digestible for everybody. Why we're interested in feral bees. I don't know if any of y'all have looked into Tom Seeley's work. He's did some studies in New York on feral populations that were far enough out that commercial operations and hobby beekeepers didn't really affect them. And what he found was that the feral bees not only were surviving better, they were thriving. The number of feral colonies were actually increasing. And we are interested in finding out why. Mapping is another large part of what we're doing. We want to map those feral populations to allow us to be able to study them better. Also with the mapping, there's a study in France where bee populations across France would have genetic drift toward a certain subspecies. And if you would take a population from one region of France and move it to another region of France, it didn't do as well. We want to map those regions in the United States, starting with Kentucky, to see if we observe that same drift between regions, to see if we can take those genetics that are good in a specific region and kind of keep them in that region and say, hey, these are the genetics you need in this region. Our avenues of research, swarm team collections, DNA analysis, morphometric. I'll go over all of these. So Apis Rescue operates a swarm team. It's an all-volunteer swarm team. Basically what we do is we operate a hotline. We run the logistics of the swarm team. And then we have local beekeepers in different regions who go out and collect the swarms. They keep those swarms in that local region. So that way we're not transporting from one region to another. We're not going out and poaching beekeeper swarms. The beekeepers getting the swarms are the beekeepers in that region. And then we go and we do research on those swarms.
Can you define region? Is that county, state? How do you do that?
Technically, right now we're going basically by club areas and stuff, but as we do the mapping, we'll redefine those regions based on what does best in areas. So the bees will actually define the regions eventually.
How are you defining, or if at all, how do you identify a feral swarm versus a swarm that's coming from one of your local beekeepers?
That is some of the research we're actually working on.
Okay.
Right now we're in kind of a data collection phase. We're collecting this data for statistical analysis, and then we hope to we hope to be able to find a good way to tell what is an escape colony versus a multi-generational feral colony.
Where are you located?
Sorry.
Where are you located at?
Me personally, or
Or
Apis
the
Rescue?
company?
Apis Rescue is, our head office is in Henry County, basically my house. But, our swarm team operates in an area about the size of Connecticut in, northern Kentucky and southern Indiana.
Is there a place on your website where you can go see the progress that you've made already, or different areas that?
Currently, no, but it is in the works. We're actually, with our mapping data, we're working with GIS to do the mapping data, and we are gonna start putting that as we have results on our website.
Is it only swarms, or do you do cutout samples too?
Generally, we don't do cutouts. Technically, in the state of Kentucky, to do a cutout from a house or a structure, you have to have a sprayer's license. We have people we refer
That's not
to.
true. That's not true.
State apers would disagree.
I went through all this when I started up my business. I've been doing removals now for about 15 years.
Yeah.
I didn't have to have any type of license before that.
What we were told by the state apers is that is the law. They don't actively enforce the law, but being a business that deals with this stuff, we don't take the risk of doing it. But we do have people that we refer cutouts to.
See, I've got commercial insurance that covers all that, so it's no problem of mine.
Yeah.
Yeah, not the illegal.
Yeah, that's the problem with insurance. As soon as you're breaking the law, they can be like, we ain't covering that. Our goal with the swarm team is eventually expand this model throughout Kentucky. Currently, it's one swarm team, but as it gets larger, we'll break that up into actual different swarm teams operated in the same way. So DNA analysis. So the swarms that we're collecting, we collect DNA samples from. Currently, we're working with Kentucky State University, or no, Purdue University to do. They have a grant where they're working to collect DNA samples, and we're sending them samples, and then we get DNA results back from them.
Do you supply the collection materials and mailers and all of that, or how does that work?
Right now, our swarm team collects a swarm. We go out, me and another person goes out and we collect data off of it. We go through a set of tests to test the colony, looking for diseases and doing mite washes, doing aggression tests and that kind of thing. And then we collect a sample after the mite wash and we send it out. However, one of the things I have in here later, we are going to start offering kits so that other people can do it as well. One of the things we're looking at when we're sequencing is Africanized honeybee genetics, seeing how much of that is in there. We've had a few issues in Kentucky last year with extremely aggressive hives, and some of the data has been showing that it's Africanized genetics are drifting in. And then one of the other things we're doing with the DNA analysis is we are confirming previously collected morphometric data. So when they look at subspecies and that kind of thing, we use morphometric data on the wing veins. We're trying to confirm that. So morphometrics is a quantitative analysis of form. So basically measurements of the body to determine species or subspecies. We use a program called Drawwing. It said it measures the veins in the wings, it measures the angles between certain ones, and can give you a pretty accurate result of what subspecies of honeybee you're looking at. Like I said, and then what we want to do is compare that with DNA analysis to confirm that what they have previously recorded is correct. And then our citizen science initiative is, these are our kits that we want to offer. We had planned this year, but they didn't quite make it. We decided to planning a conference would be a great idea, and it kind of took up all of our time. So we would offer DNA and morphometric kits to the public, which basically is a single kit. You know, you do a mite wash, you put, you rinse the bees in the alcohol for the mite wash, and you drain them out, and you put them in our test tube, and you seal that up, and it gets sent back to us. The public would purchase these kits. They send them back to us, following the instructions that we have, doing the observations that we have listed for them to do. And then we would take that and send it off and have the DNA analysis done and do the morphometric data. And then we would send them back their results, but also that would allow us to put more statistical data in our database.
Is there any kind of genetic testing that you all send bees to, or how can you tell some is feral stock?
That's one of the things that we want to look at. There's several avenues we think that we can identify feral stock, but we have to get the statistical data to be able to determine whether we're accurate on that. One of the things is like the genetic drift to specific subspecies based on the climate in the region, I think is a way to identify. I believe that bees that have been feral in an area over multiple generations will have that drift, and that we can identify them that way.
Are you using the same protocols that the Bee Informed partnership is using? You've heard of the BIP, obviously.
I've heard of it. I'm not sure about their protocols.
Oh, okay. Essentially, we're doing the same thing. It's just that they call them sentinel. We pick a colony that's, you know, within one of our yards or something like that, and we're doing exactly the same thing.
Okay.
So I would encourage you to at least
I will take a
reach
look
out
at
to
that.
them.
More people doing this kind of research, the better, because more statistical data, the more you know.
Data is good.
So we would record the data and send the citizen science scientists the results, which is kind of a little nice thing. You'd be like, oh, I've got this subspecies of bees for them, but a lot of that helps us to collect more data. Feral colony analysis. This is the analysis we go through with our feral colonies. Our first thing is coloration. We use this. This is actually for soil, but there's a lot of overlap in bee color on that. So it's just a standardized way that we can go into the colony and take a color sample of the bees, which is part of the morphometrics, you know, to help us find ways to better identify subspecies. And then we do a disease assessment. So we'll do a mite wash. We'll go through and do an inspection looking for different diseases. Of course, the mite count. And then we sample we take a sample for DNA. And then we do our colony aggression test, which is pretty awesome. I don't know if you can all see all the little white dots on here. These are all stingers. With our colony aggression assessment, we take a medicine ball on a tripod and we pull it back and we let it swing back and forth in front of the entrance of the colony. And we count the stingers, so the sting response. And then we take a 10-pound medicine ball and we drop it on the top of the colony from a specific distance 3 times. We do it over again and count the sting response.
It's a weed whacker.
Yeah.
Works just as well.
Yeah. Yeah, this one's a little more repeatable for experimentation. But this right here is a colony we caught in Bardstown, down the road here in Louisville. When we caught this swarm, I got 30 stings.
Wow.
From a swarm. So there were close to 200 stingers in that bag before we did the drop test.
Wow.
We're pretty sure they were advertised. I would have loved to have been able to collect samples off of them, but they were determined to be too much of a danger. So we split it into 10 splits and moved it to 10 different places.
When you did that decrease their voracity?
They were still very aggressive for about 6 weeks, but there were so few bees that it really helped. You didn't have to worry about it.
It diluted the genetics
Yeah,
after that.
it diluted the bees enough that you didn't have to worry about killing people, which is our main concern. But at that point, we had diluted the genetics enough that they weren't a viable sample.
What time of year do y'all do those tests like this?
Generally June or July. It varies. It's hard to schedule time with everybody and stuff. We tend to wait till after the major swarm season in the spring, which usually be it's about June. I think we did these end of June this year, last year. It's a pretty interesting time this year. This is actually, we were really excited. It was our first time going out collecting data, so. And then our origin mapping, so I spoke about already. For the most part, it's wanting to do that regional mapping, but anytime a swarm team member goes out and collects a swarm, they record the coordinates of the swarm so that we can throw that in GIS and map where everything came from, which is slightly accurate. You know how bees are, though. What we will hope learn from the feral colonies, the main is how they manage pests and disease. And then we are looking, like I said, to a better way to learn how to tell between an actual feral, multi-generation feral colony as opposed to somebody's bees that escaped last year.
Just remind, so I'm definitely not a scientist, but what do you call, you say multi-generational, at what point do you consider them feral? How many generations?
Technically, any that escaped are feral by definition, but
But in this particular
I'm interested in looking at things that have been feral for at least 5 years, which would be
You're looking for trees,
yeah,
right? You're looking for
basically.
trees.
Yeah, because what I was thinking, okay, are you calling feral bees? My bees swarm, I don't catch them, they go get a tree for 2 years. Are you kind of feral? You're saying 5 years.
I would like 5, yes.
Wow.
That's to be able to see that genetic drift, you need a few generations to track it.
Is a generation a season, or is it
Generally.
general?
Yeah. But it's based on the swarm cycle. So, yeah. Generally, with a healthy colony, they're going to swarm once in the spring and maybe once in the fall, but usually once in the spring, and that's a generation. So.
Do you ever see any feral, bees in like an old building for 15, 20 years?
Yeah.
There's a lot of that out there.
Yeah, we've got a few. We got a few that trees that have had bees in them for people say they've had them for 20 years. And it's always hard to confirm, which is another thing that this kind of research can help with, is being able to confirm that. But,
A lot of the information you can get off people who that owns a building or something that
Yeah.
sees them all these
yeah,
years.
they see them every year, and that's a very valuable data point.
Yeah, but it's subjective.
But it's subjective, exactly. So you have to be able to balance that with the research.
Objective.
I'm gonna hold presents.
And we want to understand how these feral colonies manage pests and disease. It's like I said, Tom Seeley's work is proving that they're not only surviving, but they're thriving. I want to find out whether this is a genetic factor or an environmental factor. And if it's a genetic factor, is that something we can breed into our bees, that we can bring into our apiaries to help? And same if it's an environmental factor, is that something that we can add to our hives or, incorporate into our management to help? So there's a lot of people interested in breeding bees to be, more resistant to this or that, but I'm not sure how much work has been done on, whether that's actually the factor, whether it's the genetics is the factor or the environment is the factor. how we plan on benefiting the beekeeping community. Education is a big one. Bee group presentations is something that we're working on right now. We are wanting to create education programs, accredited comprehensive education programs that can be dropped into any bee club, large or small. We have been doing some small stuff like this Henry County Public Library is a beekeeper class that I did recently. This conference is a big part of that too. We would like to bring in well-experienced beekeepers who have a wealth of knowledge to help spread that knowledge to people and to focus on the education aspect and not the profitability. I think we did pretty good at this one. And of course, presentations at conferences like this and other conferences. We did Hive Life this year. Jake, one of my partners in Apis Rescue, went to Hive Life and did a presentation. He's doing a few here today too. And we went to the Beekeepers of Indiana conference last month. Breeding programs is definitely a way to benefit the beekeeping community. The plan with the breeding programs is we determine that the factors allowing bees to survive better are genetic. We want to spin up breeding programs to help put these genetics into people's apiaries. And what we plan to do is to work with existing breeding clubs and breeding programs. So we're not reinventing the wheel. We can use their expertise. And then we want to create region-specific breeding programs. Like I said, we believe that genetic drift will vary by region, and we'd like to map those regions and then have breeding programs in each of those regions for that region. And we want to promote that local stock. You get a lot of people who want the biggest, best, and most famous bee that they can find, you know, that the YouTuber told them about or whatever. And we would like to start, and this goes back to the education as well, we'd like to start promoting those local genetics that are adapted for your area so that people are going to have more success, that new beekeepers aren't going to come in and be frustrated in 3 years and quit. We want to use the feral stock that we're looking at to create better survival stock. You know, by grafting larvae and creating queens for the breeding program. That's if the research works out, it is a genetic factor, not an environmental factor. And this is our prospective timeline. Last year, we started, like I said, collecting our DNA samples and doing our testing, which went pretty well so far. This year, we're going to continue with that. We had our conference, which I think is going pretty decent. I was very surprised by the turnout. We were expecting about 50 people. So. We are actually applying for SARE grants this year. My wife, who is a board member, is a PhD candidate, and in her day job, she also writes grants all day long. So she is writing grants for us for research.
Who's your wife? What's her name?
Aisha.
Aisha?
Now, she is technically a soil scientist, but we also, I think, are applying to a couple of grants from APCM. And we are expanding our swarm teams, and hopefully our citizen science initiative will get up and out this year. That one's still in the work. We're still working out the instructions and stuff and how we want to handle people collecting samples, because that can be, the data can be a little more subjective when you don't have controls in place. So we want to make sure that's as perfect as we can before we start shipping those out.
How far out in the region are you looking to get fabric?
Right now, we're looking at all of Kentucky and part of Indiana. Eventually, if, you know, if our research pans out the way we think, it would be nice to be able to map the entire United States. But that's,
I do a lot of tree cutouts, and I've never even thought about contacting anybody, so next time we do a cutout, I'll call you.
okay. We can get you some sample kits and so too. Those tree cutouts are very exciting to us because
They're old.
yeah, a lot of times they are. Yeah. Next year, basically more of the same. We're gonna, you know, with our swarm team and collecting samples, of course, and we plan on having our conference again. We are going to start analyzing our morphometric data next year as well, which currently all of our samples for morphometric data we have desiccated and have desiccants. They're stored with desiccant to keep them dry and in a freezer. And as we collect more samples, we're going to start breaking those down and doing measurements, doing our wing analysis, and then other measurements on the body too, just trying to find an easy way to identify subspecies of honeybees without having to run them through, you know, DNA analysis. Something that's maybe not 100%, but close. And then we're going to try to start working on the feral stock breeding program to start getting some of the connections made, not necessarily the actual breeding. You know, that's probably several years out, but making the connections with people who are operating breeding programs and clubs that are doing that to kind of start greasing the wheels to get that done.
You find very many viruses and stuff in these wild colonies?
Not a whole lot of viruses. We find mites on some of them. Some of them are pretty clean. The ones with mites occasionally will have, you know, but nothing real bad. At this point, the biggest thing we found is the Africanized genetics. That's most of the swarms that I've collected personally have actually been pretty healthy and turned into pretty decent colonies. I keep all of my swarm colonies in a special swarm yard away from all of my other, you know, personal colonies. But other than the really aggressive one, I can't think of any bad ones I've had. All right, so how can you help? Anybody wants to join our swarm team, if you're within our area, even if you're outside of our area, you can fill out an application, and when we move into that area, we'll give you a call. We do have an interview process for it. We have criteria for our beekeepers. The main one being a minimum of 3 years experience. Purchasing and submitting citizen science kits. That's always a great way to help. Continue coming to our conference. And then becoming a member in the future, something that in the future, once we have data to share, we would like to start a membership. You pay so many dollars a month, and then you get a newsletter that keeps you updated on what we're doing. I don't think we have enough data currently to justify people paying for a newsletter that's going to come out once a year, but but once we can start sending something out once a month, we're going to spin that up. Basically, that's just to help fund research. Purchasing our swag, we got t-shirts. I don't know if you guys have seen them over here. We've got Apes Rescue t-shirts and conference t-shirts. And making a donation is always great. And that's pretty much it. Thank you, guys.
The 2008 study for the morphometric, was that a peer-reviewed study, and was it published? If it was, where?
It is published. I'm not sure on the peer reviewing of it. I believe it is. It's been a few years since I've read it.
Do you remember the name off the top of your head so I can copy it out? It's on your slide.
It's pretty interesting stuff. There, a lot of the stuff on morphometrics is done in England, where they've been trying to save Apis mellifera mellifera.
That's it.
Yeah.
Tolfiski.
What's the reason for your keeping swarms in a swarm yard, more or less diseasing, mites, anything else? that you
Yeah,
don't
me personally,
Okay.
not all of our swarm team members do that, but personally, I don't want to take the chance of introducing something into one of my yards, you know, some EFB or heaven forbid, was it the paralysis?
K-wing,
Yeah, K-wing.
black virus, yep.
Yeah, there's a lot of stuff that could be introduced, and
Okay.
I operate a business other than Apis Rescue with honeybees, and I can't take the hit
Fair enough.
it's just basically just a isolation yard. So once they've been in that yard for long enough to assess them, sometimes I'll move them out into my other yards. But,
See, I use my backyard as my staging area, and then I make sure they're good to go before
yeah.
moving them.
Chris, I've had, 8 years ago, I bought two packages.
Mhm.
You know, honestly, I have no idea where they originated. I know where I bought them from, but I don't know where they came
Yeah,
from.
I'm pretty
So-
sure I know.
So, I've been operating in the same apiary this will be my 8th year.
Yep.
Are those bees pretty much acclimated? You can say, to-
I believe so, because you don't introduce any new genetics into your yard. I have high hopes that they are, and I sent off samples of your bees to have DNA research done on them. So,
Okay.
so yeah, the hope is they have acclimated to our area, and since you do open breeding, I'm very curious to see what they got. Any more questions?
What do you do with all the swarms you get? Do you help new beekeepers or do you sell them?
So
Okay.
all of our swarm members are local beekeepers from the area where they get them. We have rules like you're not allowed to take more than an hour to go get them. We want those bees to be as close to where they swarmed. And then the rules for the swarm team is that swarm has to be tracked. Any splits off of it are theirs to do with what they want. Any honey off of it is theirs to do with what they want. But the main swarm has to remain as close to where it came from, you know, in their apiaries, and it has to be tracked. So I said, anything that comes off of it, anything that's produced from it, is the beekeepers. That's kind of their payment for being a volunteer.
So then what data are you tracking then?
We do mite counts, we do the DNA analysis, and the disease assessment and aggression, and then we continue to do mite counts and stuff over several years, because sometimes you will get a swarm in that has no mites, and you're like, oh, this is a great one for a study, and then the next season it's mite bombed, and they're just covered in them, and it kills them.
Do you
And
track treatment then too?
yes, absolutely.
Do you all treat with the same thing then? So you have the same across the
We
board?
do not all treat with the same thing, but one of the requirements of swarm team members is they have to have an integrated pest management plan, and we review that pest management plan. We don't want beekeepers who are using the same treatment over and over and over
Right.
again. But
How many volunteers?
I think we have 20 right now, and we're actually recruiting right now, and I've got about 20 more applications in my email that I haven't had a chance to look at, which is wonderful for us, because we usually get like 2 a year. So last year was a interesting year with swarms. I don't know if y'all had the same problems we had, but we dubbed it Swarmageddon. We ran out of beekeepers and bee equipment to capture swarms in. And when we do that, we actually will contact the local clubs there so that they can, if they have anybody that wants stuff. So we're not interested in poaching swarms from people. We want to keep those swarms with local members.
Do you have members all over the state?
We currently only have one number. I said our main region is here. It's about the size of Connecticut, but this is kind of the epicenter. Oldham County technically is the epicenter. Apis Rescue technically started as a swarm team in Oldham County. And then when I took over as president of the swarm team, I incorporated and started up Apis Rescue. So created a board and wanted to take it a little further. So.